Thank you, Donna. Thank you very much. It is really a pleasure to be here celebrating the 50th anniversary of the Caucus of Women in Statistics. I am really thrilled to be able to be here with you and also to be, the beginning of a periodic event, perhaps. So that's the title that I decided to put, Spanish Women in Statistics, what else? And so let me tell you a little bit of what is this. My introduction, which is going to be very brief. I will tell you about the statistical societies in Spain, mainly how many members we have in each society and how many of them are women and how many of them are led by women. I will tell you a little bit of the research areas of the Spanish statisticians. And then I will introduce my three speakers, Carmen Armero, María José Rodríguez, and Amaya Iparragirre. So as you can see here, there are 5 different logos. The first 3 correspond to national associations or societies. Going from left to right, the Sociedad Española de Bioestadística, which is the Spanish region of the International Biometric Society. And then the Sociedad de Estadística e Investigación Operativa, which is a national statistics society that is quite old. It's probably the oldest of the 5. And it includes not only the statisticians, but also scientists working in operations research. And then we have BioStatNet, which is a network of biostatisticians. And I'm going to tell you a little bit of everyone. And then we have 2 local, meaning 2 autonomic, because you know that in Spain we have autonomies, like a federation, let's put this way. And then we have one which is in Catalonia, where I live, the Sociedad Catalana de Estadística, and one in Galicia, where COTE is, which is the Sociedad de Galega para la Promoción de Estadística y la Investigación de Operaciones. So starting with BioStatNet, this is a national network of biostatisticians. We started this network in 2010, so it's going to be 11 years. And we have 8 different nodes, and we already have 262 members. 121 of them are women, so we are not bad, 46%, so almost balanced. And very, very well balanced with respect to, as I say, presidents, which in fact are the PIs, because as I said, we have 8 nodes, and therefore there are 8 PIs, And one of them is Carmen Armero, myself is another one of the PIs, and then Carmen Cadarso and Maria Durban, and then the other 4 are men. So this is something that gathers the research in biostatistics that is done in Spain, and we are very active and we have a lot of ideas, and it was a very wonderful initiative that has been really good also for the young people in Spain, because now they know much better, from one region to the other. Then, as I was saying before, we have the Sociedad Española de Bioestadística, which is, as I said, the Spanish region of the International Biometric Society. I speak too fast. This is, of course, much older. We have over 250 members, 161 of men of women. So we are very good in terms of parity, because we overdone, more than men. However, in terms of presidents, we have had 14 presidents, 5 of them women, so we have to work a little bit more on that. Although the next president is going to be a woman, so we are going to be a little bit, this 35% would be a little bit higher. And then we have the oldest of the statistical societies in Spain, which is CEIO, that has also 260 members. And here the number of women is not that bad, it's 45%. However, the number of presidents among the 15, only 1 has been a woman. So I hope that this is something that in the next few years will change a little bit. Then the Sociedad Catalana de Estadística is very young. It has 10 years. But it already gathers 188 members, 73 of them are women. And among the 5 presidents, 2 of them have been women. Let me also say that between the members in the Sociedad Catalana de Estadística, Sociedad Española de Bioestadística, BioStatNet, CEIO, and Esgapello, there are many intersections. So many of us are, for instance, myself, I am in Biostatnet, in Sello, en Sociedad Catalana de Estadística, and in Sociedad Española de Bioestadística. So, it's not that you cannot add up all these members. Many of us are the same. And then the fifth one is the Sociedad Galega, it's also for statistics and operations research, and it's quite large. It has more than 200 members, and it's, as you can see, also is very good in terms of women, because 57% of them are women. This is a very nice society because it also includes, as far as I know, because I have been there sometimes, it also includes all the statisticians that are working, for instance, in high school, or just doing the promotion of statistics in secondary schools. Among the presidents, among the 6 presidents, 2 of them have been women. So this is all what I can say about these societies. And I was trying to do some research on the research areas in statistics. And in fact, this was quite difficult. And what you have here is, like a cloud of topics in which some of us work. It doesn't pretend to be exhaustive. And in fact, it has been very well based in the information we have in Biostatnet. Let me tell you before I continue that these slides, I have the luck of being helped by Amaya and by Leire Garmendia, that they helped me a lot, to put together these slides, and gave me, all these good ideas with colors and everything. So as I was saying, the research areas in statistics, there are many. And so we have a large group working in Bayesian analysis, Bayesian computation. We also have many groups in survival analysis, among them, some of them using Bayesian modeling, also doing statistical software. We also have many different groups working with longitudinal data analysis. Also, a number of groups work with splines, and also within the survival analysis, there are some people working on multi-state models, in fact, several groups as well. And then we also have quite a number of groups that work in flexible modeling, regression flexible modeling, also with ROC curves, some of them. And we also have people working in clinical trials, different types of like bioequivalence, composite endpoints, and many other things in clinical trials. And we also have a number of important of persons working on omics data, bioinformatics, so or multivariate methods for omics data, microbiome analysis. Okay, so there are many other things working on, and in fact, what I have now is like a wish, because when I was starting to prepare this talk, I mean, not this talk, just this introduction, I was much more ambitious, and I really wanted to know, how many statisticians work in Spain and how many of them are women. Oh my God, that's a very difficult problem. I mean, I think the problem starts that who is a statistician? How do you make a difference between a statistician and, for instance, data scientists? I mean, how do you decide where is there a border? Is there a frontier? And how do you count them? So that's why I went to see in the societies, that's much easier. And then something that it could be easier to if I have more time, maybe for next year, is to find out how many statisticians are in academy, because that's something that you can just go through all the universities, at least the public universities in Spain, which public universities in Spain are in general the best universities. And so probably from there, you can just go to those that have statistics departments or mathematical departments with a statistics section and check how many statisticians work and how many of them are women. Also, something that I think it would be really nice, it would be to find out how many research groups are in statistics. Again, we have this problem of how do you define statistics and which other things do you put together, like, for instance, biostatistics, bioinformatics, but still, if we can, estimate this and how many are led by women. And of course, something much more ambitious would be, okay, if you go to different work areas like academy, banks, pharmaceutical industry, consultancy, how many, what is the proportion of women? So here, I think we have a problem that it would be really nice to solve, in the sense of having these figures. And so probably we can work, maybe we have to do a survey more than trying to get all the data. And so I really hope that with my colleagues, with Carmen, Cote, and Amaya, and many other persons that could, maybe for the next few years, we could try to put together this information. Okay, so now after these 10 minutes, let me just go to introduce to my speakers. I will start with Amaya Iparragirre. Amaya is a PhD student at Universidad del País Vasco, and she works at the Biostatistics Research Group. You can see here in this beautiful picture in the beach. And she works, her research areas has, among others, the development and validation of prediction models, and also complex survey data analysis. And we thought that it was nice, and in fact, this was the idea, I think, of Carmen or Cote, that say, okay, why don't we talk about also the hobbies we have? Not, so, we're not going to say how many papers, how many sites, just. So Amaya was sharing with us that she likes to travel, she likes reading, and she likes cooking. So, I think that we can do many things together, and of course, we can try to enjoy her cooking. And Amaya will tell us in 5 minutes, she will just develop this talk about to wait or not to wait, that's still the question, on the development of prediction models for complex survey data. Our second speaker, it will be María José Rodríguez, we call her Cote. She is a Ramón y Cajal Fellow at Universidad de Vigo, that's in Galicia, and she belongs to the Statistical Inference Decision and Operations Research Group, it's called CIDOR. Among her areas of research, she works in the statistical evaluation of the diagnostic and prognostic value of clinical biomarkers, in the development of methods for flexible regression models, and also on the proposal of new statistical methods for spatial and spatial temporal processes, with a special focus on agricultural experiments. And she has also many hobbies, besides being very nice, and fun to put together this very interesting picture, which we love it. But she also likes to travel. So, Amaya and Cote so far can go together in a trip. Also reading, so we can share, which are the books that we like. Sewing, this is something I don't share, and I don't think Amaya. Being with friends, oh my God, that's something. And drinking wine. And I think that we can really be with her in all these hobbies, And she will be talking about the spatial analysis of field trials using penalized splines. And then our third speaker is Carmen Armero. She is professor at Universidad de Valencia and is the PI of the Valencia Bayesian Research Group that is called VABAR. And her main research areas are on Bayesian hierarchical models, longitudinal and survival models, and Bayesian modeling in archaeology, and many more. This is just a synthesis. Okay, so what about her hobbies? She likes, I don't know how to say it in English, Pilates, we say in Spanish. Okay, I don't think I'm gonna go with you to Pilates. She likes plants, and I also like plants. She likes reading, you see? Also, we can share books, and drinking white wine. I like that. I mean, I really want to go with this group. Okay, Carmen, thank you very much. And she will be telling us about Bayesian survival models with spatial covariance. And so I was going to finish like that, but then Carmen and Maya and Cote told me, okay, you also have to say which are your research areas and hobbies. So here I am. And I am professor at Universitat Politècnica de Catalunya. I am the PI of the research group in biostatistics and bioinformatics. And my main research areas are in clinical trials and in particular in composite endpoints, also in survival analysis and in particular dealing with complex sensory patterns and multi-state models. And about my hobbies, gardening, which is like Carmen with plants, reading. Oh my God, the four of us like reading. So I took this picture because I'm reading, traveling, and gin tonics with friends. So, these gin tonics with friends puts together, being with friends, wine, etc. So I think that having so many things in common, not only in these research areas, but also in the hobbies, I think that we can go really far. So I think that with that, I will finish my presentation. And if I know how to, okay, so I will just go try to, let's see, yeah. Okay, I'm trying to, what I'm trying to do is I'm trying to stop to share my screen, but I cannot do it. I don't know why. I'm sorry.
You did it, Lupe, I think.
You did. I think we're ready for Amaya.
I did it already? Okay, yeah. Perfect. Okay, perfect. I was not aware. Okay, so thank you very much again. And Amaya, it's your turn. Thanks a lot for being with us.
Thank you, Lupe, for the introduction. And I would like to start by congratulating the Caucus for Women in Statistics on its 50th anniversary, and also appreciating the effort that they have made in organizing this 24-hour around the world event. And also, I would like to thank in particular to Lupe for thinking of me when organizing this roundtable. I think we are lucky to have very wonderful and admirable women statisticians in Spain. So I'm really pleased to be here today with Lupe Cote and Carmen. So let's start. My name is Amaya Paraguirre and I'm a PhD student at the University of the Basque Country. Today, I'm going to talk to you about my PhD thesis project. This is a joint work with Iran Zubarrio and Inmaculada Arostegui, which are my PhD advisors. But first of all, before starting with the talk, I would like to start by introducing our research group. BioStat is a biostatistics research group, a group formed by researchers that are working in the Basque Country in the north of Spain here. And these are some of the members of this group, not all of them, but some of them. And I think that this picture depicts quite well the relevance of women in this research group. So very briefly, the main goals of this research group of BioStat are, on the one hand, the development and application and validation of statistical methodology, and on the other hand, the transference of results to society. We work with applications in many areas that can be summarized as experimental, social, and health sciences. And some of the research lines of this group are the development and validation of prediction models, on the one hand for complex survey data, and on the other hand for modeling the evolution of patients with chronic diseases. The creation of a score from multiple patient survey responses, the development of statistical methodology for modeling quality of life for patient-reported outcomes, and finally, the software development. So let's start with the talk. The goal of this talk is to talk about my PhD thesis project, and I'm going to focus in this first part of the development and validation of prediction models, and in particular on the development of those prediction models for complex survey data. The goal of the talk is to describe the objective of my PhD thesis and briefly talk about what we are doing and explain why we think that to wait or not to wait is still the question. So let's start. I think that to understand the idea of my thesis project, it's necessary to describe, first of all, these three concepts: the prediction models, survey data, and complex sampling designs. So now I'm going to proceed to describe these three concepts. So first of all, what do we do? What do we work on? Well, we work on the development of prediction models, and the goal of prediction models is to make future predictions based on known past behaviors. In other words, is to make predictions for unobserved data based on the data that have been previously observed. But in order to end up with a valid and useful model that could be applied in the future, for example, to new patients, it's necessary to follow several steps when developing prediction models. Those steps that were described by Steyerberg goes from the estimation of model parameters and the selection of covariates to the validation of the fitted models. And what kind of data do we work with? Well, in this context, we fit prediction models to survey data. When we work with survey data, we are usually interested in obtaining information about a particular population, which is of interest for the survey. In this context, this population is usually a finite population, and each individual of this finite population can be identified in some way. So first of all, this finite population is sampled following some sampling process, and the survey is carried out for this sampled units. And so new information is obtained for these sampled observations. Now our goal is to make conclusions related to the population based on the information we have obtained by means of the survey fitting prediction models. So what about the sampling process? In this context, we usually work with samples obtained based on complex sampling designs. In the literature, there are described several techniques to obtain samples based on this finite population, and the most commonly used sampling techniques are simple random sampling, stratification, and cluster sampling. And those techniques could be applied in one or more stages, sometimes combining some of them in different stages. But today, I'm going to focus on one step, stratified samples, because when we work with this kind of data, and our samples are usually obtained based on this sampling technique. So in this context, suppose this is the finite population of interest for our survey, and we want to obtain a sample based on this finite population. So first of all, different strata are defined in this population. These strata are usually defined based on one or a combination of more than two or more variables that are available for the individuals in the finite population. And then simple random sampling is applied in each of these strata. And a determined or pre-specified number of units is sampled from each of these strata. So for example, suppose that from this strata or stratum of 20 individuals, we decide to sample 4 of them randomly, and that from this stratum of 4 units, we decide to sample 2 of them randomly. It should be noted that it's not the same to sample 4 units from 20 than 2 units from 4. The inclusion probability or probability to be sampled for the individuals in this stratum is not the same as the probability to be sampled for the individuals in this stratum. For this reason, a sampling weight is assigned to each sampled unit that indicates the number of units that each observation represents in the finite population. So for example, the sampling weight for these 4 units, sampled units, is 5, indicating that each of them represents 5 individuals like themselves in the finite population. In the same way, the sampling weight for these 2 sampled units is 2, indicating the same. It should also be noted that the sampling weights for the units sampled from the same stratum is the same, but this number is not equal to the sampling unit assigned to the unit sampled from other strata. Okay, so now the next question is, well, and what is the effect of these complex sampling designs, and in particular the sampling weights, on the development of prediction models? Should we consider these sampling weights when developing prediction models? Yes or no? So as can be seen in these papers from 1973, 1984, and 1988, they started to asking this question some years ago. And in particular, I would like to mention this paper from 1988, written by Smith, entitled "To Wait or Not to Wait?" That is the question. So very briefly, summarizing very briefly all the information that we can read about this in the literature, we can say that this question can be approached from two different perspectives. On the one hand, from the model-based perspective, we can say that if the model is well specified, then it's not necessary to use sampling weights for estimating prediction models. But on the other hand, researchers that adopt design-based perspective warn about the possibility to obtain biased estimates if the sampling weights are ignored in the process. So this discussion between these two different perspectives causes some doubts nowadays among the researchers that have to work with this kind of data, and these more recent papers are proof of it. So for this reason, we can say that to wait or not to wait, that's still the question. Still, we need to do more research on this area to see whether we should or not consider the sampling weights when developing prediction models, and if we should use them, how to incorporate them in this process. So this is the main goal of my thesis project, and on the one hand, to analyze how these sampling weights affect in different steps of the development process of prediction models, and on the other hand, to propose new methodology that incorporates sampling weights if necessary. So far we have worked in the estimation of logistic regression model parameters. The goal has been to evaluate the behavior of different estimation methods that are usually applied in the literature for estimating logistic regression models that consider or not the sampling weights when estimating these coefficients for complex survey data. And we have performed a simulation study based on real survey data. We have concluded that when we don't use sampling weights when necessary, we obtain worse results than when we use them when they are not needed. In the second place, we have also worked on the estimation of optimal cutoff points for the classification of individuals, for classifying those individuals as event or non-event, and we have proposed a new methodology considering sampling weights in the estimation process of these optimal cutoff points. We have seen that these weighted estimations of the optimal cutoff points are better estimates than the unweighted ones in this context. And finally, now we are working on the estimation, estimating empirical AUC of logistic regression models in this context when we work with complex survey data with the aim of estimating, measuring the discrimination ability of this logistic regression model, considering sampling weights. So this is what we have done so far, but we think that it's important to continue doing research on this area, and we would like to continue with this work in researching how do those sampling weights affect in the rest of the steps that we have to follow to develop prediction models so that we can develop good and valid and useful prediction models also when we work with complex survey data. So here are some references that I have mentioned during the talk. And so thank you very much for your attention.
Thank you, Amaya. Very nice talk, very nice project, very interesting project. And we are looking forward to all your results and to a very nice future as a statistician. And so now our next speaker is going to be Coté Rodriguez. And so Coté, whenever you want, you can share your screen. And you could also, you are silent, so you should just unmute yourself.
Okay, now can you see my screen? Okay. So thank you very much. So I would like to start thanking Lupe for inviting me to be part of this session to celebrate the 50th anniversary of the Caucus for Women in Statistics Society. It is really a pleasure to be today here. And it is also really a pleasure to share this time and to share this session with three amazing women who I admire both professionally and personally. So it's really, really a pleasure. So thank you very much, Lupe, for the invitation. When Lupe invited me to be part of this session, I had to decide the topic I would like to talk about. So I decided to talk about the work I have been doing for the last years on the analysis of field trial experiments. This is a project, this is a work I am particularly proud of, because although from the statistical or the methodological point of view, what we have proposed, it's nothing really complex or fancy. The truth is that the work has had a rather big impact, and it is currently being used not only by researchers on the field, but also by breeding companies. And for an applied statistician like myself, seeing a work in which you have participated and collaborated being used and being useful, it is extremely rewarding. So that's why I decided to present this work. And of course, this is not a work that I have done alone. So let's see whether, okay. But, well, many people have been a part of it. And here you have some of them. Okay, this is, I would say, a rather interdisciplinary team as needed, with different but also complementary backgrounds. And among all of them, I would like to especially mention Diana Perez. She is a statistician from Colombia, and she's currently a PhD student at the VAST Center for Applied Mathematics here in Spain. And I hope I will have time to present part of the work that Diana has been doing. She has been working on the analysis of high throughput phenotyping data. And I hope I will have time to present her work at the end of this talk. And well, whenever I have to present my work on the analysis of field trial experiments, I always start with this slide and the next one, which I hope will give some background about what we are talking about when we speak about the analysis of field trial experiments. So, well, the aim of a field trial experiment, it's usually to evaluate the effect that a genotype or a series of genotypes, or if you prefer crop varieties, have on a phenotype of interest. So for instance, the phenotype can be the yield, or perhaps it can be the resistance to a particular plague, or even a quality measure. And although not always, in most cases, the aim of a field trial experiment is commercial. So the final objective will be to determine those genotypes that perform the best according to the phenotype of interest, to further commercialize these genotypes, these varieties, as cultivars. Well, but however, I mean, it is well known that in the expression of a blind phenotype, the environment plays a huge role. So for instance, the fertility of the field where the experiment takes place may have, and actually will have, an effect on the yield that is obtained, which is not related to the performance of the genotypes, which is, as I say, the aim of the experiment, the aim of the study. Okay, so when analyzing a field trial experiment, the phenotypic expression due to the environment needs to be accurately separated from that due to the genetic, which once again it's the aim of the experiment, the aim of this study. And here you have an example of a field trial experiment. This is a grid trial conducted in Chile. You have more information about the experiment and the aim in the paper by Lava and colleagues. And in particular, in this experiment, a total of 384 genotypes were shown in a field of 40 rows and 20 columns. So we have a total of 800 plots or observations. And as usual in this kind of experiments, all genotypes were randomly allocated to the plots according to a particular design. In this case, an alpha lattice design with 20 incomplete blocks, each containing 20 different genotypes. And then all genotypes were shown at least twice, that is, all genotypes were replicated. And finally, the trait of interest or the phenotype of interest here is the grain yield. And here in this plot here, you have the observed grain yield at each plot in the field. And as you can see, there is a clear but also a strong spatial pattern, kind of northeast, southwest. However, due to the randomization, but also the presence of replicates, in principle, this pattern cannot be attributed to the genetics, and it is probably a pure environmental effect. Unfortunately, most of the times there will be no covariates to relate this pattern or to relate this effect to. We have no information about, for instance, the fertility of the field. So modeling strategies must rely on the location or the position, the spatial coordinates of each plot in the field. Okay, so as I said, so the aim here is to explain what we have observed in our example, the grain yield, in terms of the genotypes. But this model here, it's very simple and it is not realistic, but because we need to correct for the spatial variation. And in this setting, for the analysis of field trial experiments, the proposal by Gilmore and colleagues in a paper in 1997 has become the standard modeling strategy, especially among applied breeders. And it was also the starting point of our work. And in this paper, the authors, what they propose is to decompose the spatial variation in three components, or more precisely, they speak about three sources of spatial variation. So the first one is what they denote the extraneous variation. And this is related with the experimental procedure. So for instance, caused by the way the field was prepared before or during sowing or planting. So this is really a systematic effect. There are usually covariates to relate this effect to. And for field trial experiment, it is quite common to incorporate into the model the random factors associated with the rows and columns of the plots in the field in order to account, let's say, for the way the machines work. Okay, then we have the second source of variation, the second source, of spatial variation, which is the global trend variation or the large scale variation. And this is usually modeled by means of additive functions, possibly smooth, defined in terms of the spatial coordinates, which I denote here as x1 and x2. And then we have the last source of variation, which is the local trend variation or the small scale variation. And here the authors propose to model this source of variation by means of a spatially dependent stochastic component, xi. And it is quite common here to assume a separable autoregressive process of order 1 in the row and columns. So as I said, this is the traditional approach due to Gilmore and colleagues. I will say that this is a really powerful and useful approach, but it also has some drawbacks. So in this setting, what have we proposed then? Well, what we proposed was to explicitly, but also jointly modeling both the global and the local trend variation, in this case by means of a two-dimensional interaction smooth surface defined in terms of the spatial coordinates, x1 and x2. So note that this approach avoids the need to model the local trend variation by means of a correlated noise, which in some cases might be far from trivial, and it also presents it may present some problems. And well, in more detail, what we propose is to model and estimate this two-dimensional smooth surface using anisotropic two-dimensional penalized splines. Well, this work led to two publications. The first one with a more methodological or statistical component, where we present more or less in detail our approach, and we also derived some results, some interesting results. And then a second one where the focus was put on a particular application. And here, the first author, Julio Velasco, a PhD student at the time, did an incredible job because he analyzed hundreds of field trial experiments, and then he compared the performance of the new approach, our approach, and the traditional approach by Gilmore. And the conclusion was that our approach was rather competitive. In some cases, it was better, but perhaps most importantly, it was easier to use, at least for a routine analysis of field trial experiments. And I think that one of the reasons, or perhaps the major reason why this approach has become, I would say, rather popular, it's because along with these two papers, we also developed an R package called SPATS, Spatial Analysis of Trials Using Splines, which can be freely downloaded from the grant repository. And I really believe that this is what made a difference. So apart from proposing something which has a really good behavior, also to give the tool to use it in practice. And I think that this work really made a difference. And here you have an example on how to use SPATS for the analysis of the trial experiments that I presented at the beginning of this talk. So the main function of the package is called SPATS. And here we have to specify the response variable of interest. So that is the phenotype of interest. In our case, that's the Joule. Then we need to specify the name of the variable in our data set, which corresponds to the genotypes. Then we need to specify whether we want to model these genotypes as fixed or random. We are in the mixed model framework. Then we need to specify the spatial component, that is the two-dimensional smooth surface. I'm not going to go into the details, but since we are using penalized splines, we need to specify the nodes for X1 and X2, the spatial coordinates. Then through argument random, we specify the random components to be included into the model. For our particular example, we include the random factors associated with the columns and rows. And finally, through argument fix, if needed, we could incorporate some extra fix effects. For this particular example, there are none. Well, as usual in R, we can obtain some numerical results using the summary function. I'm not going to go into the details, but we can obtain results in terms of variances. Again, we are in the mixed model framework, or in terms of effective dimensions. This is for us easier to interpret, but also we can obtain some graphical results. And these are the 6 plots that the package provides. So here we have the raw data, then we have the fitted values, we also have the residuals. Here we have the fitted spatial trend, that is the estimated two-dimensional smooth surface. And finally, here we have the genotypic predictions, which is really what we are interested in. So for instance, if the aim of this experiment is or was selection, that is to select the best genotypes, we could now just focus our attention on, let's say, the 10% of genotypes performing the best, and then to further study them. So what I have just presented allows us to analyze a field trial experiment, but for a particular situation in which the phenotype of interest was measured only once, usually at the end of the study. However, with the arrival of high-throughput phenotyping platforms, we now have access to long-time series of repeated measurements of the phenotype of interest between seed emergence and physiological maturity. This is an example of high-throughput phenotyping data. And this is the kind of data that we now have. So this video here shows, hopefully it works, the evolution over time of the spatial distribution of the phenotype of interest. In this case, this is the leaf area for the 35 times at which this particular phenotype was measured. So the range is exactly the same for all time points. That's why at the beginning, we do not observe any kind of variation or variability. And then we see that as the time passes, we start to observe how the plants, how the replicates start to differ. Okay, the white areas denote the missing data. And this is another way to look at the data. So in this case, what we have here is the evolution over time, that is the trajectories for the plants of the phenotypic data, the phenotype of interest for the plants belonging to 2 different genotypes. And as you can see, it seems that the plants here, the plants belonging to this genotype here performed better than this one. So they grew more and perhaps they grew faster. So the aim here in this case, it's as before to be able to estimate the genetic signal, but now we would like to be able to estimate the evolution over time of the genetic signal. But once again, and as before, we need to account for, we need to correct for now the evolution over time of these environmental factors, the spatial variation. So it means that for the analysis of high throughput phenotyping data, what we need are spatiotemporal models. And also the computational aspect here, it's quite a challenge because we may have hundreds of thousands of observations, but also highly parameterized models. And this has been the topic of Diana's PhD thesis. And very recently, she has proposed a two-stage approach for the analysis of this kind of data. Here you have the paper, and also here you can download, if you are interested in the code needed to reproduce all the results shown in the paper. So in the first stage, a SPATS model, the one I just presented, it fits to the phenotypic data, but separately for each measurement time. And then the phenotypic data is corrected by considering or only considering the source, the estimated source of variation, which are of interest, which is what we call here the spatially corrected data. And note that this spatially corrected data do not show any, at least clear, spatial pattern, which is otherwise present here in the raw data. And then in the second stage, the focus is on what we are really interested in, on modeling the genetic signal as a function of time for the corrected phenotype that we obtain in the first stage. And here we use a p-spline hierarchical curve data model, which allows, among other things, to obtain the evolution over time of the genetic signal. These are these orange lines. I'm not sure whether you can see them. So the blue lines will be the evolution over time, the estimated evolution over time for the plants. The orange are the evolution over time for the genetics, for the genotypes, what we are really interested in. And based on these curves, we could in principle extract some time-dependent or independent characteristics for the different genotypes, and they can be useful to compare the performance of the different genotypes. And just to finish, I think that one of the things that really makes this research area very exciting and interesting is that the list of things to be done is really large, it's huge. So here you have some of those areas where we are currently working on, or at least where we would like to work in the near future. Not all, and that's all from my side. So once again, Lupe, thanks a lot for the invitation. It has been really a pleasure. And I would like to finish just saying that if we really want a healthy science, then diversity at many different respects, it's really a must. So that we should work on it. So thank you very much, Lupe. Thanks to all of you.
Oh, thank you, Cote. Very nice last slide. I want it. You have to send it to me.
I will.
Thank you for a wonderful talk. I really enjoyed it. And you know, I bet that you are happy with this topic, not only because it's exciting that you know that you have been able to put together many of your work before, so that this can be applied to something that is being done, but also because I think, I mean, that's something that never happened to me. You know, it's like you are just doing things, those things that founders of statistics did, right? Because working with designs in agriculture, I mean, it's just going to the basics, okay? It's going to the beginning. Oh my God, that's really something. That's really amazing. So I'm sure that, and the other thing that is funny is that I guess that there are not many professions in the world that says, oh, we are so happy. We have so many things to do. We have so much work to do. Oh
it's overwhelming at some point. Like, I don't know, there are many things to do. Which one should I try to address first?
But you see, you know, not many other professions are so happy of having so much more work to do. Okay. Anyway, thanks a lot, Cote. And now we are going to give the screen to Carmen Armero, our third speaker. So Carmen, whenever you want, you can share your screen.
Okay. What is happening here? Sorry, I don't know. Okay, but I don't know with the, okay.
Can you try Control L? Can you try Control L and to have the full screen? If not, it's fine. I mean, don't worry if you...
Okay, it's okay. I don't know what's happening. Oof. Okay, I try. It's a pleasure to be here, even this situation of with slides. I appreciate a lot the role, the paper of the caucus for women in statistics, and because it's try always to help women in statistics, and this is a part of the long road to be free and to be equal. No? And also, I would like to thank Lupe for inviting me to participate in this very nice meeting. And also it's a pleasure to share this meeting with Maya and with Cote, because you are wonderful women and wonderful statisticians. Okay, I would like to speak a bit about the Bayesian survival models with spatial coverage. This is a joint work with Virgilio Gómez-Rubio from the University of Castilla-La Mancha, Fran Llopis and Gabriel Sanfelix from Sabio, which is a research center in Valencia dedicated to medicine, and Dorota Moenarczyk and Pere Puig from the Universitat Autònoma de Barcelona. I would like to present the group of research in Bayesian statistics. The name is PAVAR, and we are a lot of people that with a route very clear. This is the University of Valencia. There are some people that continue working in the University of Valencia, but other people has gone, and but we continue in connection. We continue working together. We have, Anabel, Antonio, me, David, and Gabriel and Miguel. All these are in the University of Valencia, but we have Gonzalo and Virgilio in the University of Castilla-La Mancha. We have also Joaquin now in Valencia. In the other university, the competing is sometimes enemy university, the Polytechnic. We have Javi in the University of Elche. We have María Eugenia in Madrid, in the Rey Juan Carlos, and also Stefano in Madrid. But in the Carlos III, we have Alicia in the University of León, and we have Elena, who is working in an institute dedicated to agriculture, and has the same interest as Cote, and also Blanca in Fisabio, and also Facu. He is in France, but he is working in agriculture issues also. We have Danilo in Chile, and we have the master, who is Jim Berger. Okay, we are a group that our research is basically on Bayesian statistics, and basically we have 3 blocks of research. We are people working in longitudinal and survival models. There are people working in spatiotemporal hierarchical models, and there are people working in more methodological issues that are model checking and model selection. I would like to speak something about about survival models in which we are working. We have done something about the basic survival models. We have studied something about curation models. We have dedicated a lot of time in analyzing the baseline hazard function in modeling different elements with some regularization issues, and also to use bugs with different survival models. Also, we have done something in joint models of survival and longitudinal data, and this has been a very interesting work with Lupe. I remember that we started with a real problem, and we were two teams, the frequentist team with Lupe and the Bayesian team, and we decided to analyze the problem. The frequentist with the frequentist tools, the Bayesian with the Bayesian tools, and then we share the science and the wine, of course. And this was a very nice time because it was very fruitful, and we did a lot of things. And now we are starting to work with survival, but with also putting some spatial elements. We have started analyzing the risk of stroke in Poland, and now that is in progress. It's not finished. It's only we are working with illness death models. We are putting some coverage, some spatial coverage, in order to assess the hip fracture in elder populations. A bit about the risk of stroke in Poland. Poland is here in the middle of Europe, and we were interested in studying the prevalence of stroke in this country with regard to individual demographic information, administrative information. This was the counties, the powiats in the language of in Poland, some socioeconomic information and pharmacological information. We had a lot of data, 500,000 of people from the Polish National Health Fund. And we started to analyze the main survival time. That was the time in which an individual who lives in a powiat will suffer an ischemic stroke. In terms of coverage, the random effect, and the measurement error that was expressed in terms of a Gamble distribution. Here, this is the first time that we use the Leroux model that combines the neighbor matrix and an over-dispersion element. We complete the specification of the Bayesian model with the prior distribution. We consider a simple scenario with non-informative prior distributions and prior independence. And because we had a lot of data, we know that the MCNC were very slow, and then we use ILDA. It was more fast. Here we have some results. This is for men, this is for women, this is 3 different ages, and we are representing the posterior mean of the probability of a stroke. And we appreciate that the age, as we know, is a risk for the stroke, and men are less protected for the stroke. And here also we appreciate some spatial distribution, and we know that this part of the Poland and this part has the most higher risk of the stroke. Now we go to the Valencian community. I know that Lupe is here, Amaya is here, Cote is here, I'm here. And now we are interested in hip, to study the hip fracture that we know that is a real problem in elder people. We have a lot of data from a long period of time. We follow up people, all people, 65 or more. that has been a fracture and has been discharged alive after being in the hospital. We are interested in basically 3 events. The starting event that is fracture, to have another fracture, and death. The relevant times are time to fracture, to refracture, to refracture, to death, fracture, to death. Of course, we can put a second refracture, a third, but at the moment it's okay. We were very interested in not only these times, but also in assess the transition probabilities. And here we have some results for the posterior mean of the transition probabilities between fracture here, fracture to death in women, in men. And here we have a person that in state at the time 0 is in the initial state is fracture, and here is the evolution for going to death. Okay, and we know that women has less smaller probabilities of death than men. Okay. On the contrary, this is from fracture to refracture, and here we have that women refracture more, okay? And this is, this second line is from refracture to continue in the refractory state, and here is from refracture to death. Obviously, this is the sum, this probability and this probability, the sum is 1, okay? Because there are the total probabilities in the line. Okay? And it seems that the epidemiologists think that there can be some structure, some spatial structure in these transition probabilities in these times, and then this is the thing that we are doing. We are trying to put some spatial relationships between in the different times, and we think in something that is characteristic of the regional unit, in this case in the health department, and something that is related with the neighbors of. And then we are thinking about the structure of this element and these elements, because there are a lot of possibilities. And I think that is okay. And when we have more information about this, we will show you. But I think it's time to finish. And okay, these are some reference and some thank yous. And okay, and I'm sorry for all this. Okay, thank you very much.
Thank you, Carmen, for such a nice talk. I think it's very interesting. I didn't know about this hip fracture. That was one of your last topics. And so I'm really interested because we're also doing things in multi-state modeling, and I'm sure that...
No, I know.
You know. Okay. I mean, it's so nice to have so many things together. I mean, it's so...
Yes.
Yeah. So thanks a lot, Carmen, and thanks to the 3 of you for such a wonderful talk. Each one of them has been really nice, and they are really interesting. And thank you for your willingness to participate. You always say yes, so you are really nice guys. You're nice people, and so thank you for making the statistics in Spain much better. Such a much better place to be just because of people like you, and also I think that this, as Cote was saying, this diversity that we also it's not only a gender issue, it's much more, is bigger, is larger than gender issue. So Cote's slide was really good for that, and so that's what we want, that every person and every minority or non-minority is is represented with all of us. And I think that you represent that. And I think that that's, I really want to thank you, because people like you, make the atmosphere so much nicer, so much better, that at the end, I think that I can say, we are colleagues, that's for sure, but we are friends. We like to share statistics, but we also like to share white wine. I don't have any problem, I can have white wine, red wine, gin tonic. And Amaya is a little younger, but she also likes to have all that. And also, she will be doing the cooking, and we will be putting all the wine. So thank you very much. And let me end, just thank you, the Caucus of Women in Statistics, for such a nice opportunity that we have been able to share some of the statistics that we do in Spain. You have seen only 3 instances, So you all now that we are very good in many things, and in particular in Bayesian analysis, and in complex survey data, and in modeling, in flexible modeling, and also in applications. We're applying this in many different fields. So I think that it's a good opportunity for we as women and as statisticians in Spain, that people know that we're just doing very interesting things, that we have fun when we do all these things, and that we really want to continue just sharing this with all of you. So thank you for this opportunity, Donna, in behalf of